Hello, my name is Nari Beatty, and today's date is June 27, 2022. So I'm going to present to you my case connections um, responses, and I chose to respond to cases 1 and 2. So starting with case 1, it says pyruvate kinase catalyzes the rate-limiting ATP generating step of glycolysis, in which phosphoenolpyruvate is converted to pyruvate. Multiple isozymes or isoenzymes of pyruvate kinase exist in mammals, type L, which is found in the liver and kidneys, type R, which is expressed in erythrocytes, type M1, which is found in tissues such as muscle and brain, and type 2, which is present in self-renewing cells such as embryonic and adult stem cells. So the first question for case 1 states, under normal conditions, describe all the ways in which pyruvate kinase is regulated in the cell. So my response to question 1 for case 1, I talked about how under normal conditions, the way in which pyruvate kinase is regulated in the cell is or through feed-forward regulation and covalent regulation in the liver. So during feed-forward regulation in the liver, pyruvate kinase is activated by fructose 1,6-bisphosphate, which is made through the phosphofructokinase, or PFK1 reaction. The feed-forward regulation process connects increased levels or produces increased levels of fructose 1,6-bisphosphate, which then soon activates pyruvate kinase. And then, in addition to the feed-forward regulation, we have during covalent regulation in the liver, CAMP-dependent protein kinase, or PK, drives phosphorylation and soon leads to the deactivation of the hepatic isozyme of pyruvate kinase. This phosphorylation and deactivation of pyruvate kinase only occurs in the liver and is driven by low blood glucose levels and elevated intracellular CAMP concentrations. So, as a result of this, phosphoenolpyruvate, or PEP, isn't able to proceed with glycolysis and enters gluconeogenesis instead. And then we result with dephosphorylation of pyruvate kinase through phosphatase, which is an enzyme that occurs and it allows pyruvate kinase to be reactivated. So that was my response for question 1. And transitioning to question 2 of case 1, it says that many types of cancer cells are predominantly glycolytic and overexpress the oncoprotein MYC, which is a cancer-expressing cell. And that, interestingly, the classical oncoprotein MYC has been found to promote preferential expression of pyruvate kinase type M2 over pyruvate kinase type M1. So the isoform M2 is characteristically found in a low activity state and is ineffective at promoting glycolysis when compared to pyruvate kinase type L or type M1. So the question for number 2 states, this logic seems counterintuitive for a glycolytic tissue. What is the rationale for this mechanism? And what other pathways may cancer cells be diverting substrate to? So my response to question 2 basically stated that within cancer cells, it's common for type pyruvate kinase M2 to be increased. So as type M2 is triggered by the oncoprotein NYC, which is the cancer-expressing cell or gene, glucose transporter 1 or GLUT1 is expressed and is connected to the proliferation or growth and development of these cancer cells. So as a result of that, the glucose uptake is spurred or stimulated, as well as the process of glycolysis. So now I will transition into case 2, and the question for that is, a 16-year-old female is recently diagnosed with a deficiency of muscle glycogen phosphorylase. She and her family are concerned and ask the following questions. Based on what you know about skeletal muscle glycogenolysis and muscle metabolism, please respond to each question with a thoughtful answer that describes the metabolism in these various scenarios in language that your patient will understand. So the first question is, I'm not sure I understand the issue, the scenario that I just stated about the patient. Can you explain how muscle glycogen is used normally during exercise? So my response to question 1 is that during normal exercise, glycogenolysis occurs primarily as glycogen synthase and glycogen phosphorylase are regulated to restore the body's ATP source. Next, the glycogen synthase and phosphorylase are allosterically managed by the effector molecules, which are basically glucose and glucose 6-phosphate, to meet the ATP needs of the skeletal muscle in this case. So when the muscle is being exercised, glucagon or epinephrine, which are supporting hormone, will serve as signaling hormones to increase the blood glucose levels and provide ATP needs for the skeletal muscle. So moving on to question 2, it states, I really like to take long walks. Will I still be able to do this with my friends? My response to that is, with your deficiency of glycogen phosphorylase in your muscles, I don't think it would be a good idea to walk for long periods of time. In regards to that, there is a disease called McArdle disease where lack of this enzyme can lead to muscle being incapable of utilizing glycogen as an energy source, eventually causing muscle fatigue and pain within the muscle. The third question states, as a result of this deficiency, will I need to get up at night to eat or maintain my blood glucose levels? My response to that is, waking up at night isn't necessarily needed to maintain the blood glucose levels, because your body will be signaled and produced. It is signaled and produced when your glycogen stores or degradation occurs, and it can also be produced through dietary intake of various starches, so monosaccharides, polysaccharides, everything of that sort. However, dietary intake of these starches may not be the most dependable or primary way to restore one's blood glucose levels if they are that low. And that is because in some cases the sources of the starches may not always circulate consistently through the blood. So when your blood glucose drops in the absence of that dietary intake, your muscle tissue will produce glucose through glycerol, lactate, pyruvate, and amino acids as carbon sources for that ATP metabolism to restore or regenerate the skeletal muscle. And then lastly, the patient asked, do I need to worry about producing excessive lactate during these intense anaerobic exercises? So I read that overproducing amounts of lactate during anaerobic exercise is not extremely harmful, but excess amounts could possibly lead to an increase in pH levels within your skeletal muscle and could disturb other useful metabolites to contribute to muscle wasting. And that is the end of my case connections presentation. Thank you.